Hello students, have you wondered about the term polysaccharides? And actually today we will discuss in our Physio-Pulse channels the polysaccharides and the different types of polysaccharides, with their structure as well as the application or significance. Now let us understand what is polysaccharides. Actually we already discussed that when monosaccharide is a single sugar and disaccharide when two monosaccharides combined with the glycosidic bonds they'll form the disaccharides, and after that oligosaccharides comes 2 to 10 of the carbon molecules, and then whenever it is more than 10 monosaccharides or above 10 monosaccharides, then it will produce some polysaccharides. So there are these are the polymerized products of the many monosaccharide units, that is called the polysaccharides. So they may be two types. One is homoglycan, which is composed of a single kind of monosaccharides. It's the same type of monosaccharides like the starch, glycogen, cellulose. We all know. But secondly, heteroglycans, that are composed of the two or more different monosaccharides, like the hyaluronic acid, even also chondroitin sulfates. So let us understand one by one polysaccharides, their structure as well as their applications or significance. First of all, starch, very important term, starch. The structure is that it is a reverse carbohydrate plant kingdom, and the source is potato, and this is the cereals, rice, wheat, and other food grains. They can found the starch molecules. Starch is composed of amylose and amylopectin. When starch is treated with the boiling water, 10 to 20% is solubilized, and this part is called the amylose. And amylose is made up of glucose unit with the alpha 1,4 glycoside linkage to form the unbranched long chain, and with the molecular weight that is the 4 lakhs near about the Dalton, or more than this molecular weight. Now the insoluble part of which is absorbed of water and forms the paste like gel, that is called the amylopectin. So we already know that the amylose and amylopectin both two are present in the starch. Amylopectin is also made up of the glucose units, but it is highly branched with a molecular weight and more than 1 million. So the branching point alpha 1,6 linkage made up with the amylopectin. So this is 1,4 glycoside linkage with the amylose, and amylopectin is 1,6 glycoside linkage. So when it is hydrolyzed, what will happen? Starch, we know that starch is a blue, will form a blue color complex with the iodine, and this color disappears on heating and again reappears after cooling. So this is the sensitive test for the starch. So starch is non-reducing because the free sugar groups are negligible in number. So it can say that it is a non-reducing sugar, and when starch is hydrolyzed with the mild acid, smaller fragments are produced. So thus the hydrolysis of a for a short time produces the amylodextrin, and even also which gives the violet color with the iodine and which is non-reducing. So further hydrolysis produces the erythrodextrin, which gives a red color with iodine and mild reduction in Benedict solution. So these are the structural differences in the starch amylodextrin, and even also erythrodextrin. Now the action of the amylase is on the starch. What are the actions? Actually, physiological actions: the salivary amylase, pancreatic amylase, are the alpha amylases, which act at random on the alpha 1,4 glycosidic bonds to split the starch into a smaller unit, that is called the dextrin, and finally alpha maltose. Beta amylase are of plant origin, like the almonds and also germinating seeds, and which split starch to form the beta maltose, and they act on the amylose to split the maltose unit consecutively. And thus the enzyme starts its action from one end. When beta amylase acts on the amylopectin, maltase are liberated, and at the end the branches of the amylopectin, until the action of the enzyme is blocked at the alpha or 1,6 glycoside linkage. So the action of beta amylase stops the branching point and leaving a large molecule, and which is called the dextrin or residual dextrin. So in this way we can say the function, actually the structure as well as the important functions of the starch molecule. This is an so now in second homoglycans, if we have to see the already we know the starch, and then second is the glycogen. Glycogen is a reserve carbohydrate in animals and it is stored in the liver as well as the muscles. About 5% of the weight of liver is made up of glycogen. So excess carbohydrates are deposited as glycogen, and glycogen is composed of glucose units and which joins by the alpha 1,4 links with the straight chains. So it also alpha 1,6 glycosides linkage also at the branching point. So molecular weight of glycogen is about 5 millions, and innermost core of the glycogen contains the primer protein. So glycogenin or glycogen is that is more branch and more compact of the myelopectin. And number three, that is the one another important polysaccharide is cellulose, and it is the supporting tissues of plants. Cellulose constitutes the 99% of cotton and 50% of wood, and is the most abundant organic material in the nature. So it is made up of the glucose units combined with the beta 1,4 linkage, and it has a straight line structures with no branching points. The molecular weight is 2 to 5 million. So beta 1,4 bases are hydrolyzed by the enzyme cellulose, and that is by this enzyme but this enzyme, when it is absent in the animal and the human digest system, the hence cellulose cannot be digested. So this is very important term. That is the cellulose bias. That is that this type of enzyme is absent in the animal and human digestive system. So hence cellulose cannot be digested in the human body. Now the herbivorous animals have a large cecum and which harbor the bacteria, which are the bacteria, and this bacteria can hydrolyze this cellulose and glucose produced utilized by animals. So cellulose has a variety of commercial applications, as it is the starting of material to produce the fibers, cellulose, nitrocellulose, plastics, even etc. So in this way, cellulose are also very important in the human life. Now another important that is the inulin. Inulin is a polymer, or one type of polymer homoglycan composed of deep fructose unit repeating beta 1,2 linkage, and it is the reserve carbohydrate present in the various bulbs and tubers such as the dahlia, then this type of garlic, onion, etc. And it is a clinically used in the renal clearance value or renal clearance test, and also moreover it can use to determine the glomerular filtration rate. So inulin specifically uses for the kidney test or kidney renal clearance test, and after that we can measure or calculate the glomerular filtration rates. So dextran is another type of polysaccharides. These are the highly branched homopolymers of glucose units 1,6,1,4,1,3 linkage, and they they are produced in the microorganism. They have a molecular weight 1 million to 4 million, and since they will not easily go out of the vascular compartment, they are used to intravenous infusion, plasma volume expander treatment, and hypovolemic shock. And lastly, another important homopolysaccharide is the chitin, that is present in the exoskeleton of these insects, and it is composed of the ineacetyl glucosamine with the beta glycosidic linkage. So these are all the homoglycans, and because of that, they are made up of a single kind of the polysaccharides, types of sugar molecule. They are linking with the polymerizations of the chain. And another important what is called the heteroglycans, and these are polysaccharide containing more than one type of sugar residues like the agar, and very important for your practical aspects as because it is prepared from the CUETs and contains the galactose, glucose, and other sugar. It is dissolved in water 100 degrees Celsius, which upon cooling set into the gel. Agar cannot be digested by the bacteria, hence used widely as a supporting agent to the culture bacterial colonies. Agar is also used to supporting medium for immunodivision and immunoelectrolysis. Both very important for our practical aspects. And agarose is made up of the galactose combined with the 3,6 anhydrogalactose unit. It is used for the matrix of the electrophoresis. So in this way, we all discuss about these homopolysaccharides and heteropolysaccharides. And besides this, there are some mucopolysaccharides present that we will discuss separately in our next lectures. So students, I think we all have discussed detail about the polysaccharides and different types of polysaccharides with their structure as well as the clinical and physiological functions. So these all will help you for your upcoming exam and even also competitive exam or any other any other higher classes for understanding these polysaccharides. So thank you for watching this lecture. Hope this session will help you to understand the topic clearly. If you found this video useful or helpful, please like, share and subscribe to our Physio-Pulse. Do not forget to press the notification bell so you never miss our upcoming lecture like biology, even biochemistry, physiology. And if you have any question, any suggestion, please give some comments in our comment section. And thank you for watching, and we'll see you in the next lecture. Until then, keep learning.